So first of all, I would like to thank the organizers of the COST webinars for the invitation. It's really a great opportunity to present our research here. So the study I'm going to present you was conducted as part of my master's project when I was in the group of Denis Duboul at the University of Geneva, and some of the work was also achieved at the EPFL in Lausanne. Now, there is obviously no need to make a long introduction on genome structure to this audience, but let me just set the ground for our study. So as you all know, mammalian genomes are organized at various size scales, from nucleosomes to chromosome territories, with important implications for genome function. Now, among this range of structures, we distinguish intermediate size chromatin segments called topologically associating domains or TADs, which are self-interacting regions of the genome that often bring cis-regulatory elements and their target genes together into close spatial proximity. Now, in my master's project, I have been focusing on chromatin loops and TADs as features of interest with respect to genome organization. But for the moment, let me briefly remind how loops and TADs are formed. So to date, the prevailing model to explain the formation of chromatin loops and TADs is the so-called loop extrusion model involving the architectural proteins CTCF and cohesin. And according to this model, the cohesin complex pulls or extrudes a stretch of chromatin and stops when it reaches bound CTCF sites, preferentially when they are in a convergent orientation, thereby forming a loop. Now, TADs can be understood as an accumulation of loops among a cell population, whereas each of these independent loops can vary at the single cell level. And importantly, the limits between TADs, which are known as TAD boundaries or borders, prevent most chromatin contacts from going across them and are enriched for CTCF sites displaying this type of configuration. And while this holds true genome-wide in Denis' lab, we are particularly interested in the TADs and boundaries of the HoxD gene loci. So HoxD genes are found within clusters at 4 different loci, from HoxA to HoxD, in both human and mouse. And this brings me to our model system, the HoxD locus, which is depicted here. So very briefly, the HoxD gene cluster itself sits at the border between two TADs that we call the T-DOM and the C-DOM. Now, apart from this gene cluster, this region on the right, called CS3840, forms a weaker border that segregates the T-DOM into subdomains. Importantly, region CS3840 comprises 3 highly conserved non-coding sequences, CS38, the limb enhancers CS39 and CS40, as well as 3 CTCF binding sites that are all sharing the same orientation. And in the present study, we first demonstrated using HiC that upon deletion of region CS3840, the 2 subTADs of the T-DOM merge to form a single and more homogeneous domain, as you can appreciate on this HiC heat map. So based on this observation, we asked whether the formation of this boundary would be context dependent or not. In other words, if we were to place this region somewhere else in the genome, would it reconstitute a boundary there? In order to answer this question, we generated a construct of 45 kb called TgN 3840, comprising the entire region, and performed random transgenesis by pronuclear injection. Subsequently, because we absolutely needed to know what happened at the level of the integration, we characterized the resulting transgenic line extensively using various approaches, which I will go through today. So we started characterizing the line by a technique called targeted locus amplification, or TLA. So as a brief summary, TLA is a method that is very similar to 4C-seq. However, due to specific differences in the restriction and the library preparation workflow, TLA produces larger hybrid DNA circles and results in a better sequencing readout of the so-called hybrid junctions or connections, which are connecting DNA fragments of different origin. Now, the key is that the connection between the transgene and the chromosome should be present somewhere in these hybrid junctions, and this could be resolved by TLA. So we performed TLA in transgenic samples, and this is how the data look like genome-wide. The viewpoint that was used in this case was located in the transgene vector, and this peak that you can appreciate here corresponds to the endogenous region CS3840 at the HoxD locus, since the data were mapped onto the wild-type genome. Now, importantly, only a single other peak was detected elsewhere in the genome, likely reflecting the transgene integration site. So let's have a closer look at this peak. This is the 80 kb region of chromosome 10, where the integration was assigned, and you can appreciate the strong peaks in the TLA coverage for both the vector and the CS38 TLA viewpoint. Importantly, by now assessing the sequence composition of the hybrid reads located within these peaks, we could characterize one of the integration breakpoints with a base pair resolution. This revealed a connection between part of the CS38 element and chromosome 10, and therefore we concluded that at least one partial fragment of the transgene got integrated there. However, it remained unclear whether other and maybe complete copies of the construct were retained next to the partial fragment as well, and also the left integration breakpoint remained completely uncharacterized at this point. So to assess how many copies of the construct were present at the level of the integration, we performed qPCR and control freak analysis on genomic DNA. And to make it short today, I'm going to skip the control freak part and present you the results we obtained for the qPCR only. So we performed qPCR on genomic DNA samples of 3 different genotypes: wild type, heterozygous for a deletion of the endogenous region CS3840, meaning had the HoxD, and samples that were heterozygous for our integration. Now, the value along the y-axis represents absolute allele counts. So first, and as expected, no particular changes between genotypes were observed for a control region located within the HoxD cluster, HoxD8D9, and also the heterozygous deleted samples validated the quantification approach. Then concerning regions CS38 to CS40, I need to mention that an absolute allele count of 2 had to be assigned to the endogenous region. So this means that CS38 appeared to be present somehow in more than one copy at the integration, whereas CS39 and CS40 were both present in only a single copy. So in order to validate these findings and to map the missing integration breakpoint, the left breakpoint, we performed long-read nanopore sequencing, or MinION, after a release by CRISPR-Cas9, and this is a technique referred to as nCATS. Now, because conventional nanopore sequencing is generally associated with a high error rate and a low coverage genome-wide, we wanted to enrich the region of interest prior to sequencing it. And to do so, we chopped the construct out from genomic DNA in vitro using 2 distinct combinations of CRISPR guides, thereby releasing overlapping DNA fragments ranging in size from 10 to 23 kb, which we then pulled together and sequenced. Now, I have to mention at this point that based on both the TLA and the qPCR results, we assumed that the integration consisted of 1.5 copies of region CS3840 in a head-to-tail tandem configuration. So this is how the MinION output looks like in the form of a dot plot. On this, each of the dots represent a 20 base pair match between the reads and the reference, which is the assumed mutant configuration shown on the top. Now, in horizontal, you have the position along this configuration, and in vertical, the position along each MinION read. As you can see, with only 5 reads, we could map the entire integration from one end to the other. And in particular, as you can appreciate by the continuity of these oblique lines, the MinION fully validated the proposed mutant reconstruction, including its head-to-tail configuration. Now, furthermore, MinION also enabled us to characterize the left integration breakpoints, hence completing our base pair map of the TgN 3840 integration. So to summarize, we know with a good confidence level that our integration makes 63 kb in total and comprises 4 CTCF binding sites that are sharing the same orientation. Now, going to the biology, since it is now clear that the binding of the architectural proteins CTCF and also cohesin has the potential to generate boundaries in the genome, we postulated that if our transgene would act as a boundary, it should normally recruit both of these proteins. To assess this, we performed chipmentation against CTCF and the cohesin subunit RAD21 in E12.5 mouse limbs. And by doing so, we observed a recruitment of both proteins on the ectopic region 3840 with profiles that were very similar in their shapes compared to those obtained for the endogenous region of the wild type sample. Then we actually wondered whether the elements of the transgene would be able to interact with the host chromatin landscape and if this would impact on the local interactions. So this is how the host landscape looks like in wild type limbs. It is characterized by this 1.2 megabase TAD displaying a relatively low density of CTCF sites, a couple of H3K27 acetylated regions, and only a single protein coding gene called BTG1. So just briefly, we know that the transgene landed here in the right half of the TAD. So first, to score contacts established by the transgene, we performed 4cSig using viewpoints located either within the CS38 or CS40 elements of the construct. And by doing so, we could detect interactions that were mainly restricted within the limits of the welcoming TAD with maximum contact frequencies towards CTCF sites located within this TAD, among which those of the domain's boundary. Now, taking kind of the opposite view, we ask whether the interactions established by these surrounding sites would be somehow affected by the integration of the construct. And indeed, we observed that CTCF sites located at the left and right borders of the TAD now established new loops with the elements of the transgene, and that their interactions with regions located beyond the integration site were decreased in the mutant as shown here in red compared to the wild type in blue. So based on this observation, we asked whether the host TAD was maybe disrupted by the integration of the construct, and hence we perform high CON samples that were homozygous for the integration. And by doing so, we indeed noticed the reconstitution of a relatively weak boundary at the level of the insertion, causing a splitting of the host TAD. Now, interestingly, we noticed that some interactions between regions located across the integration site were maintained, and this is similar to what was observed at the endogenous region within the HoxD locus. So in other words, to summarize, these data demonstrate the reconstitution of what can be called a sub-TAD boundary in the new context. So we then set out to determine whether the integration and the structural remodeling of the locus had an impact on the expression of the BTG1 gene. To answer this question, we performed in situ hybridization of BTG1 in E12.5 embryo. So in the wild type, we observed a strong expression of the BTG1 gene in limbs, among other tissues, but in the mutant, the transcript levels appeared to be decreased, and these results were reproducible between two independent experiments. So drawing from these observations and to get some insights into a potential mechanism for the downregulation of BTG1, we went back to the 4cSig data and related them to H3K27 acetylation coverage. Of particular interest, one of the two strongly acetylated regions of the host TAD precisely matched with this viewpoint at the right TAD border that we called CTCF right. Importantly, we previously observed a substantial loss between this region and the BTG1 gene upon integration. And hence our working model is that maybe the communication between a putative enhancer located at the right TAT border and the BTG1 gene would be impaired by the integration of the construct, possibly as a consequence of the TAT splitting. However, a complete validation of this hypothesis would of course require further evidence, typically the proof that this region is a bona fide enhancer of the BTG1 gene. So now, if you would like to check it out in more details, our story is available in bioRxiv. And with this, I would like to thank all members of the Dubourg laboratories, with special thanks to the people who are involved in this project, as well as our funding agencies. And thanks very much for your attention. Now I will be very happy to take any questions.
Yeah, I was wondering, how do you actually map your reads in your transgene for the high-C experiments? How do you know that you get signal there at the insertion site and not at the endogenous site?
Yeah, it's a very nice question. Actually, what we did is, in particular, the high-C and the chipmentation of CTCF and RAD21, we worked with embryos that were brought in on top of a deletion of the endogenous region CS3840. So we could completely exclude the possibility that some reads would correspond to the HoxD at chromosome 2.
Okay, makes sense. So in this case, do you expect that maybe the deletion of the endogenous could have an indirect effect on your?
You mean on the contact at the integration site?
Yeah, I don't know.
Yeah, it's an hypothesis, but we are what I need to mention is that so the HoxD is located in chromosome 2 and the integration site in chromosome 10. So we kind of take it. We took that for granted that there wouldn't be probably not much of an influence from a deletion located in chromosome 2 for the new contacts established in chromosome 10, especially that we are looking now most likely at CTCF mediated interactions, and to my knowledge, it hasn't been shown thus far that such interactions can actually occur between chromosomes. But this is an interesting question, of course.
Okay, that's fine. Thank you.
Maybe I can ask one quick question before we move on. So I was a little bit confused because I thought that the CS enhancers were actually limb enhancers. So I could imagine a scenario where your transgene is actually ectopically activating BTG1, although it is endogenously, normally also expressed in limbs. But why don't the enhancers help promote its expression in limb?
Yeah, so this is a very, very nice question, of course. It was also one of our hypotheses that maybe, as you said, the integration of this, the CS39 enhancer, which has activity in the limbs, would activate BTG1, maybe also at earlier stage where it was shown to have activity, but we didn't observe such a trend as shown here for in 9.5 embryos. Now we think that there are different kinds of explanations to these observations. Typically, we basically observe only a very few contacts between the transgene in general and the BTG1 gene itself. I need to mention that the transgene is strongly interacting with a CTCF site that is very close to BTG1, but it's not the BTG1 gene unit per se. And there is so the contacts are low. And there is another possible explanation is that our enhancer is maybe not active in the new context, and this, but we don't know the exact answer to this question.